All right, everyone, welcome. Today, we're diving into one of the most fundamental topics in biochemistry, enzymes. They're basically the microscopic engines driving every single thing that happens in our bodies. We're going to break down how they work, how we can control them with drugs, and maybe, most importantly, how they become these crucial clues in diagnosing diseases like a heart attack. So, let's get right into it.
So here's our game plan. We'll start with the absolute basics, what enzymes are and what they do. Then we'll get into the nitty-gritty of their speed, which we call kinetics. After that, we'll look at how we can pump the brakes with inhibitors. This is super important for pharmacology. And finally, we'll connect it all to the clinic, seeing how these tiny molecules in a blood test can tell us a huge story about a patient's health.
Okay, so first things first, the catalysts of life. Let's really get to the heart of what these amazing molecules are and why they're so essential. I love this quote because it just perfectly captures the essence of an enzyme. They are the true workhorses of the living world. They're catalysts, for sure, but they're not like the ones you find in a chemistry lab. They are incredibly specific, unbelievably fast, and remarkably accurate.
So the official definition is a biological catalyst. Now, to put that into perspective, think about this. If you wanted to break down a piece of meat with acid in the lab, you'd need to boil it for days. Well, enzymes in your stomach do that exact same job in just a few hours, and they do it at body temperature. That's the power we're talking about.
And where does all this magic happen? It happens at a special little pocket on the enzyme called the active site. Now, people used to think it was like a rigid lock and a key, but we now know, thanks to Koshland's induced fit model, that it's much more dynamic. It's more like a glove molding itself perfectly around a hand. The enzyme actually changes shape to create that perfect, snug fit with its substrate. And it's important to remember, enzymes rarely work alone. Many of them need a non-protein partner to help out, which we call a coenzyme. And this is exactly why your B-complex vitamins are so important. Vitamins like niacin and riboflavin get turned into essential coenzymes like NAD+ and FAD, which are absolutely critical for making energy.
Now, enzymes are neatly organized into 6 major classes based on the type of reaction they catalyze. This might look like a lot to memorize, but don't worry. There's a really simple mnemonic to remember them in the official order. Just think OTHLIL, which stands for over the hill. That's oxidoreductases, transferases, hydrolyses, lyases, isomerases, and ligases.
All right, so we've got a handle on what enzymes are. Now let's talk about how fast they can go. It's time for Section 2, Kinetics, or the rules of speed.
There are several things that can act like a gas pedal or a brake on an enzyme. How much enzyme you have, the temperature, the pH, they're all critical. Just think about how a high fever can throw your whole body out of whack. But for our purposes, especially for understanding how the body regulates itself, one of the most important factors is the concentration of the substrate.
Okay, let's understand this step by step. To really talk about how substrate levels affect an enzyme's speed, we need a specific value. And that brings us to a super important concept in enzyme kinetics, the Michaelis-Menten constant, which we just call Km.
Now pause for a moment and think about this. Vmax is the absolute top speed an enzyme can work at, its red line. The Km tells you the exact amount of substrate you need to get that enzyme working at 50% of its top speed. And why is that so useful? Because it gives us a perfect measure of the enzyme's affinity or its attraction for its substrate.
This is the absolute key takeaway, so let's make it crystal clear. It's an inverse relationship. A low Km means the enzyme has a high affinity. It binds to its substrate really tightly and doesn't need much of it around to get going. On the flip side, a high Km means low affinity. You have to flood that enzyme with a lot of substrate to get it to work efficiently. So, if these enzymes are so powerful and efficient, how do we, and how does medicine, keep them under control? Well, the answer is inhibition. Let's talk about how we pump the brakes.
This slide is absolutely crucial for understanding how a huge number of drugs work. Let's break it down. With competitive inhibition, the inhibitor molecule looks a lot like the normal substrate. It literally competes for the same parking spot, that active site. This means you need more substrate to outcompete the inhibitor, so the Km increases. But if you add enough substrate, you can eventually overcome it and still reach the top speed, so the Vmax is unchanged. Now with non-competitive inhibition, the inhibitor binds to a totally different spot on the enzyme. It's not competing. It's like it lets the air out of the enzyme's tires. The substrate can still bind, but the enzyme just can't work as well. So the affinity for the substrate isn't affected. The Km is unchanged, but the overall top speed, the Vmax, decreases.
And this is exactly why all that kinetic stuff matters in medicine. Look at this list. So many of our most common and effective drugs are just enzyme inhibitors. Statins, which lower cholesterol, competitively inhibit a key enzyme. Allopurinol, for gout, does the same thing to the enzyme that makes uric acid. And penicillin? It works by irreversibly shutting down an enzyme that bacteria need to build their cell walls. This is biochemistry in action, right in the pharmacy.
Okay, for our final section, let's bring all this theory to the bedside. How do we actually use our knowledge of enzymes to figure out what's going on with a patient? So here's the question. How can a simple blood draw reveal something as major as a heart attack? Well, the answer is all about leakage. See, most of these enzymes are supposed to live and work inside our cells. When those cells get damaged or die, they break open and spill their contents, including their unique enzymes, out into the bloodstream. And that's where we can find them.
But we can get even smarter about it by looking for what we call isoenzymes. Just think of them as different models of the same car. They all do the same basic job, say, drive, but one model might be a sports car found in the city and another might be a truck found in the country. By figuring out which model of the enzyme is in the blood, we can pinpoint which tissue or organ has been damaged.
So, when we suspect a heart attack, or an MI, there are a few key things we look for in the blood. The classic enzyme markers are creatine kinase, or CK, and lactate dehydrogenase, LDH. We also measure cardiac troponins. Now, they're actually proteins, not enzymes, but they follow the same principle. They get released when heart muscle cells die, so they're an incredibly important marker.
And here's where those isoenzymes become so powerful. See, you can have high total CK levels from working out your skeletal muscles, but the CKMB isoenzyme is very specific to the heart muscle. Finding elevated CKMB in the blood is the earliest, most reliable enzymatic sign that a heart attack has occurred, typically showing up within 6 to 18 hours.
The LDH isoenzyme pattern gives us another fantastic clue. Normally, in your blood, the level of the LDH2 isoenzyme is higher than LDH1. But because your heart muscle is packed with LDH1, after a heart attack, all that LDH1 leaks out and the pattern flips. When we see that LDH1 is now higher than LDH2, what we call a flipped ratio, it's a classic sign confirming that there's been cardiac damage.
All right, let's do a quick quiz to help lock this in. Can you recall which elevated enzyme is a very sensitive marker for chronic alcohol use and can also point to liver issues like obstructive jaundice? I'll give you a moment to think. The answer is gamma-glutamyl transpeptidase, or GGT.
And there you have it. We've gone from the molecular dance at the active site to the numbers that define how drugs work, all the way to a simple blood test that can literally save a person's life. Enzymes are the perfect example of how fundamental biochemistry is the bedrock of clinical medicine. Keep making those connections. Great job. You've just mastered another key concept in biochemistry.